Hello, Combi Nation. My name is Subhi Siddek. I've spent over a decade in medical device, pharma, and combination product development. Our industry feels complicated sometimes. Drugs, devices, clinical trials, submissions, sterilization, validation, design control, risk management, market access, reimbursement. The list goes on. My goal is mastery, so this podcast is to ask questions I have to people who may have the answers. Each week on the Combinate Podcast, I talk to someone about their area to further understand and simplify. Whether you're a pharma person trying to understand the next wave of products, or a device person trying to navigate a pharma system you're unfamiliar with, or a newbie in both areas, I invite you to listen, and together we can simplify by combinating. Hi, everybody, and welcome to another special episode of the Combinate Podcast. I'm your host, Subhi Siddek. On this episode, we are graced and honored once more by Devin Campbell. He's the founder and managing director of Prodct, Know You. Welcome, Devin.
Prodct, Know You. That's good.
Yeah, thanks for coming on. So actually, during the last podcast, I had told you about my burning desire, and I've actually had a few topics recently that I'm just like, I don't know why I haven't talked about this yet. We're basically 150 episodes in. And one of the big combination product topics is ophthalmic devices. And basically eye injections, eye products, IV drops, etc. I hadn't talked about it until recently. From the same vein, IVDs, huge topic, and companion diagnostics. So that's what we're going to cover here today. Maybe if, as we're getting started, Devin, talk about your experience with IVDs, because it's obviously a pretty substantial part of your career.
Sure. So I started in the medical device arena in the mid-90s, and I've been there since. Almost all of my career has been in diagnostics, toward the end in life science tools, but with diagnostic capabilities eventually. The vast majority of my work with very large diagnostic players and pharma players, and then also with small startups, a lot of experience in the diagnostic space and in the IVD space. If it's unclear to anyone what we mean when we say that for in vitro diagnostics, it's where we're doing any diagnostic testing on samples removed from the person. We're generally not doing diagnostic testing like an X-ray. We're not taking the lungs out to X-ray them. We're a bone out. We leave the bone in place and we take a look at it with imaging. That's not IVD. IVD, we take it out. So we're usually working on tissue or on blood, serum, any different samples that come from the body, and to be able to take a look at them and then diagnose whether they've got particular cancers or if they're appropriate candidates for particular drugs or certain treatment regimes. That's the whole field of IVD.
And you highlighted a little bit, but a lot of your experience was in big pharma companies.
Yeah. So what's interesting, in a lot of IVDs, you don't really separate yourself too much from the big pharma side of things. So I've got background both at Roche and at Novartis, and working in the diagnostic space, a lot of people don't necessarily think of pharma players as being diagnostic players, but they absolutely are. Some of them have both sides of that coin. Roche, for example, has a strong diagnostics and device presence, but also has some pharma presence. But then others, which may seem more traditionally pharma and drug development focused, a lot of times those companies also rely on IVDs, whether it's their own or ones that they develop or ones that they work with other people, to be able to help in the drug development process, in finding particular patients, to be able to do particular testing they're trying to look at, but then also more importantly, to do like the whole promise of precision medicine and companion diagnostics, which are two subtly different things, but very similar. It's an important tool. And it's a vehicle through which a lot of those pharma players can get their tests onto the market when, let's say 20 years ago, they might not have been able to because the side effects, let's say, were too bad for a larger set of the population. And you didn't have really a good way to take those folks and say, okay, you guys will not have a bad reaction to the drug. So we'll qualify it for you, but everyone else, you guys will have bad reactions. So you don't get it. Once you take the test, you can't take the drug. So yeah, it's my experience and my background has been evenly balanced between the pharma side doing IVDs and then the true big player diagnostic and startup device companies developing IVDs.
So maybe just level set us on the different kind of the big terminology buckets that are used when it comes to this space. So IVDs we just talked about as a test, basically a test method that
Mhm.
uses a sample of some kind of biological sample from a human.
Right.
And you mentioned the removal of the sample and using the test to an end. And what about companion diagnostics and lab developed tests? Because those are also, because we just went over companion diagnostics, I think, as a qualification, disqualification, but if you could just go a little bit deeper into it.
Yeah, that's pretty much it. In a very simple nutshell, the diagnostic test is a companion to the drug, or you could think of it the other way. The drug has a companion with the diagnostic test. And in order to be prescribed the drug, you have to either you either need to be ruled in or ruled out based on whatever you're looking for. It could be genetic sequencing that we're looking for. And we have some sense that people with particular sequences may or may not respond to a particular drug, right? So that's really like the companion diagnostic side, which is different than precision medicine. Precision medicine is using the in vitro diagnostics capabilities again to really understand aspects of your system so that there's so many different drugs you could take and so many different therapies, especially think like oncology, right? Precision medicine is big in the oncology space because it gives us tools that we didn't have several decades ago. It gives us tools that we didn't have to be able to really hone in and make sure that you're getting what we believe is the best therapy for you at the time. So those are companion and precision ways to look at the IVD space.
It sounds like companion diagnostics, there's a validation that happens to them, whereas precision medicine is a tool that filters you towards a certain therapy area. Is that fair?
Yeah, you can think of it that way. In the companion side,
Can precision medicine fail? It sounds like companion diagnostics can fail to meet
it
its
can fail
intent or not.
precision can fail if it gives you, if it incorrectly tells you, oh yeah, you've got this much protein load, or we detected this maybe you have some ultrasensitive protein detection system, right? To be able to look for small amounts, you can fail by not finding it. And then you're not giving the information the doctors need to make the test. But you don't necessarily go through the whole clinical validation that you might necessarily do. That's more that you would do for a companion diagnostic or just any normal true IVD, right? So just normal true IVD that goes through regulatory pathways. We have to be able to demonstrate not just that it's safe and effective like you would for a normal traditional medical device, but we also have to prove that it is appropriate, like clinically proved, that it can pick up the biomarker that we're looking for. And that biomarker does have some primary endpoint, some clinical implication. And we say, yeah, if we say this, we can reasonably diagnose you with that, right? If we see this in your tissue sample, then we can say it's definitely a particular class of breast cancer or something like that. Those are the places where you do not want to make a mistake.
And I know we can probably do a whole episode, and we probably should, on lab developed tests, LDTs, but if you could define what that is.
Okay, so let's talk about, there's a couple different ways that you can get an IVD to market currently, right? The traditional route is to go through the FDA. I guess these are both traditional routes. The more rigorous route is you go through the FDA and you design and you develop your entire IVD, your medical device, following 820 and soon to be the QMSRs. You just go through like the normal routines that you would for a medical device. We have a little bit of extra implications on how we do the clinical testing toward the end. Then you get the approval or the clearance or the authorization, depending on which regulatory pathway we're going, and you bring your product to market, and then you can sell it to anyone who wants to be able to deliver it. There's a couple different vehicles. We can get into that later about how IVDs are delivered to patients. There's another way though. So you're referring to an LDT, which stands for, if folks don't know, a lab developed test. And so there are a huge number of independent labs all over the country, and I'm speaking just US wise, but there are equivalents even abroad. All of these labs that design and develop tests using equipment that they bought, and they come up with their own tests. They verify and validate their own tests, but using CLIA, CLIA, as the kind of governing quality system that drives like how do they do their testing and how do they deliver results and how do they control results and make sure that a specific result is associated with a specific person. It is very far, in my opinion, from the quality systems that you would expect out of 820 or the coming QMSRs.
CLIA stands for?
CLIA is Clinical Laboratory Improvement Amendments, CLIA.
And so those are the two pathways. There's the independent labs who use CLIA, and there's a more rigorous kind of FDA route that uses the QMSR.
But the sticky wicket with an LDT is that if you develop an LDT, you can't go and just sell it to, let's say some small lab develops a really cool LDT that seems to work pretty well. They can't go sell it to a big giant reference lab and do it all over the country. That big reference lab has to design and develop their own. So what happens is across the spectrum, we have dozens, if not in some cases, hundreds of different LDTs designed and developed and verified or validated by different labs, all testing the same thing, all delivering the same result.
And so
they do it poorly.
Well, I'm just trying to maybe draw let's talk LDTs later. Let's not jump into that because anyway, so what
I'll leave listeners with, an LDT route is a route to bring an IVD to market. It is not an FDA, it's not under FDA regulation at that point. It is, but they turn a blind eye to it. That is changing. So the FDA has finally, after quite a while, come out and said, you know what, we're going to start pulling these under the IVD definition, and you're going to need to bring yourself to those kinds of standards that we would expect to be able to help address some of the discontinuities that you see from lab A versus lab B versus lab C. And there's a whole world of conversation. We can go into that, but I think it's a really good thing.
Let's level set on IVDs first, and then we can go down that hole. So IVD development, right? We went through your kind of background in the last podcast that we did. You landed in product development consulting. Your background is in engineering. The IVD development process, is it completely different from normal medical device development in the user needs, design input, design output, V&V, etc.?
I would say it's not terribly different. It looks like, smells like the same thing. We're following the same processes, right? It's still 1345 and or 820 that govern us. The difference is on the upfront side, when we're thinking through user needs and design inputs, you have maybe a broader population of people that you have to think about. And because it's not just like the patient who has the knee transplant, right? There's just like you would for a normal, like an implantable medical device, or a Band-Aid's a medical device, right? So it's just a little bit of a broader spectrum that we have to think through. And so you might have a bit more on the user needs side, and you're going to have more on the design input side. Your risk side also has a ballooning space, because not only do we have to think about normal risks that you would think about, like harm to the patient while they're using the thing, or harm to the technician while they're touching something and using something. We still have to think through biological and toxicity, or if there's something you have to soak, like with saliva, you got to put it in your mouth for even just a few minutes or even a few seconds. We still have to do that level of testing, just like you would for any patient contact testing. But the different part is that in the IVD space, we are delivering very important information about the patient. And if that information is wrong, depending on the risks and what's going on and indications for use, there can be very large implications to that information being wrong. So we have this additional like layer on our user needs and our design inputs, and especially in our risk matrix. And as like all those things are intimately intertwined, where we have to think through what are the implications of if we gave the wrong result. So what could we do that might screw up our limit of detection, our LOD, or affect sensitivity or affect specificity, like our ability to be able to make the right call for the patient. And if we don't have those thought through, the implications are bad. So it's the same plus a little. We can say it simply that way.
But okay, so you just highlighted an added layer of risk management where it's like type 1, type 2 error, the risk of it showing a false positive, a false negative, and the fallout of it. Like,
yeah. So when we
how
get classified by when we go through an FDA classification process, you're looking at risk compared to a normal medical device, right? Where we're looking at safety and efficacy of the device. Here, we also have to look at the implications of making the wrong call.
But let's say it's a diagnostic where it's a positive, negative. Yes, you have this disease. No, you don't.
Mhm.
And what would be like, how far out would you go in terms of your risk management? Would you say, for example, if the test has a false positive and the person is actually a healthy patient without the disease, it's just that it gave the false positive. Therefore, we're giving, like, would you evaluate all the different courses of treatment and say giving a healthy patient this course of treatment would potentially yield this side effect? So S3 related to that
So
kind of failure.
we do look at false positives and we look at false negatives. We have to look at both. Really dependent on what we're trying to test for. But in most cases, the false negative is the more important one. The false positive,
Is that right?
yeah. So if we indicate that you've got something and you actually don't, they might repeat the test. There might be other pieces of information that the doctor is taking into consideration before she or they are making their final determination of your course of treatment. It's rare that the IVD stands alone, right? So you might get a result and say, you know what, that doesn't quite jive because that it just doesn't make sense. Maybe there was like a lab mix up or something. So they might retest it. The bigger problem is you go in suspecting something's wrong. And so let's use a good example, like PSA, right? So PSA, for prostate specific antigen testing. So PSA tests, you can get a lot of false negatives. You can get a lot of false positives. And so it happens. So what are they going to do? Well, now we're going to do some imaging. And based on the imaging, if we see something, now we're going to go do a biopsy. And then when we do that biopsy, we'll say, okay, and which is going to be another IVD test, by the way. So you just went from IVD to imaging to IVD. But you do that other IVD test on actual tissue, and then you decide, oh, this is a benign thing you have to worry about, or this is real prostate cancer we have to worry about, right? If you think through that same example, and you start off with, you go in suspecting there might be something wrong. Maybe your PSA is elevated. You go in and they do a test and they say, what? Or actually, I guess the PSA being elevated would be that first test. They go in and PSA is not elevated because the test was wrong, not because the PSA wasn't elevated. Now you've got potentially prostate cancer brewing and you're moving along without thinking anything is wrong. So you miss the opportunity to intervene. So you can see like the downstream ramifications of making that call wrong early in one case can have really big deleterious effects. And in the other case, where we've said, you've got something, let's do some imaging, let's do some more biopsies, let's do some secondary stain to better understand like what kind of cancer someone might have. That extra testing is going to happen before you actually intervene and take a drastic step with your therapies.
Yeah. In other words, if there's a test that comes back with some detection of the antigen that you talked about, you would ask questions like, okay, well, where is it specifically and how big is it and how do we target it and what drugs do we use? And all those questions will eventually, you'd have to see it before you treated it basically.
Right.
Okay. So I think that makes a lot of sense. You mentioned limit of detection. Like
Mhm.
in my experience, LOD, LOQ, sensitivity, those kinds of things, linearity, they're more typically used I'm just pulling from my experience. Those are more drug analytical method validation approaches, whereas on the device side, typically you use things like gauge R&R and stuff like that. And
Mhm.
so I guess, can you paint the picture of like the validation of the method itself and how that works?
Well, you're going to have like normal engineering verification. You'll have like device verification and validation against your, in that same order,
User needs and design input.
against your design inputs, and then against your user needs, right?
Yeah.
Whether you choose to capture it as a user need or not, there are regulatory needs of you as a device manufacturer developing IVDs. And you do have to go through, and there's like a different kind of clinical hurdle that we have to be able to pass. And a lot of that includes, it's the same sorts of things. Like, what is our limit of detection? How far down can we go? How many parts per billion of blank do we know that we can reliably measure? Because we know that something slightly above that limit of parts per billion of thing that you're looking for is where becomes clinically significant, right? So we need to have like our LOD. And when we're in certain situations, specificity and all those other terms, they still apply, right? But we have to go and get real world clinical data. We can't just do like bench testing, which is one of the areas that kind of helps separate it from normal verification validation. So we have to think very carefully through our targeted population and our indications for use. When would we use this specific test? Who's going to use it? What kind of patient population is it? And we have to be able to prove not just that it's safe and effective across that patient population, but that even in very young people and in very old people and across any demographic that you think would be within your indication for use, we need to be able to demonstrate with appropriate statistical validity that our test is capable of picking things up across that population. Otherwise, you got to carve them out in your indications for use.
Undoubtedly.
I think some tests, not all IVDs work for adults and kids. Some work just on kids, some work just for adults, and sometimes there's a kid version and an adult version. Because the children, like really little kids, their biological expression is going to be different than that for an adult. So one test can't be the same for both.
Yeah, I think where I was heading was like, whether it's design verification or an analytical drug method, you typically do some kind of test method validation.
Mhm.
Here with IVDs, the product is the test method. So where you would typically do, let's say, for example, container closure integrity testing on a vial.
Mhm.
Typically you would you'd laser drill these tiny holes into the vial to see what is my limit of detection on a CCI leak.
Mhm.
And that would be an input into how you go about testing that specific product configuration.
Mhm.
I'm just wondering that I'm sure that when they're going through the design and development process, pre-design verification, they're validating their test methods for sure, I imagine, in the IVD space, just like everywhere else. But at what point does the LOD, I don't know if you do LOQ as well. At what point does I don't know if test method envelope is the right, basically you need to set up the envelope where you're able to perform the test, right? And you
Mhm.
mentioned some of those parameters, but I'm wondering, does that happen once you've had a design that's passed VNV and then you do a final TM, like basically, okay, we've confirmed that this design meets its design intent. Now let's make sure that the test actually tests.
No,
You know,
that would have been an input into the design and development process, not just for the device itself, but let's expand it a little bit. Because remember, the device is a test, so there's often chemistry and reagents and biologicals involved, right? So it's rarely just a piece of hardware with some software. There's always like
yeah,
chemistry involved as well. And so that definitely influences the design process and how much error budgeting we allow different aspects of the system to have. We would not develop the system and then work on that afterwards. The procedures that we use to measure things be validated, sure, just like all the equipment would be calibrated and everything would be maintained and inspected. But the IVD, the test itself, right? You're going to go through and run huge sample sizes across the disparate population that's in your space. And that's going to help give you some of those numbers. Like in this space, we are able to detect the things at some frequency that we are aiming to detect. And that goes into our label.
That goes into the label.
Yeah, it goes into the labeling for the device.
And okay, so would you ever establish a minimum limit of detection, or would that just be performance based?
It depends. So if you have a breath test, right, and you had some clinical guidelines out there that said you need to run from this part per billion to this part per billion of whatever breath biomarker you're looking for in the breath, then you better damn well make sure that your system that you're developing hits that minimum that they're looking for clinical significance in the field. You might choose to go a little bit lower if you want to, and that's fine. But we need to make sure that even at very low levels that we have the right sensitivity that we can pick that thing up. If we can only pick it up once it's as high as several hundreds, but maybe the limit of detection needs to be down in the single digits part per billion, you've missed the boat. So it definitely it's a design input into the process in that case. But if you're just trying to characterize your system and understand where your LOD is, because you know that you're way below, you're way in a safe zone, then you could do that after the fact and go figure that out. But it's not part of that's not going into what the test is being used for.
Meaning if part of maybe in other words, meaning if the limit of detection is an important consideration, sometimes it falls into the design inputs, other times it may not. I'll tell you what I gathered from what you said. What I gathered from what you said is that like you mentioned the breathalyzer test, in that case, you need to be able to test a certain, there at some point, the device is useless if it can't test a certain limit.
Right.
Whereas for some tests, you know that the test is effective, but you need to have some kind of coverage to say this test won't test below whatever.
Right. And then you would carve that out. So,
and you would just say, if you have a virus load below a certain level that typically wouldn't be associated with a patient that has, but it may be, you may have such a low load that it's beyond the 99.9%. This is,
and if you're, say, let's say it's a COVID test, right? And if your COVID test is incapable of picking up that really low viral load, then it should report out, not that you're zero, right? It should just say it's an indeterminate test, right? Like we can't, or it's your viral load is undetectable or something like that, right? You don't want to give that number.
You don't want to say it's negative.
And if let's make up some numbers. If we know if your LOD is at 100 whatever units, but clinical significance is like 1,000 and above, so we know it's way down here, right? Then what if you measured on someone 80? Do you report that? You really shouldn't, because you know that you can reliably measure down to 100. So you might choose as the device manufacturer to say, if we get anything below 100, if we measure anything below 100, we're not going to share that number with you, even though we measured it. But we're not going to share the number because we know that statistically there could be some noise at the very bottom of that curve. And so instead, we're just going to say something like below 100 parts of whatever your unit is, or say under, it's too low.
But you wouldn't say it's negative.
You wouldn't say it's negative, and you would never give them a number when you're below what you know you can reasonably measure. And you would have huge amounts of data to back that up.
Is there any, what is the typical timeline for an IVD? My perception is that they would be quicker than a normal device, but I could be completely off.
Oh, that really depends on your regulatory pathway. If it's a 510K approach, it's going to be faster than if it was a de novo. So it varies a lot based on your regulatory path. I would say
but regulatory aside, like getting to the point where you're ready to submit, what about that? Or the submission requirements would be different whether or not it's de novo, so it would impact the development.
Again, it's really too variable to paint the entire IVD space with one brush, only because it really depends on like your indications for use, your use population, what you're trying to measure, the risk profile of the thing that you're trying to measure. All of that's going to influence maybe not the development cycle as it's going to influence development cycle, but let's just, if we were just to say all development magically happened in the same amount of time, right? To the point where you're ready to start clinical, your clinical variability could be dramatically wide, huge, based on some studies could be enormous and very expensive, and some studies could be very small and pretty precise and easy to do. So, and then the time associated with all that could be dramatically different. It's hard to put a number on that. I think with the smaller startups that I do a lot of work with, it's consistent that people are surprised how long and how expensive the clinical testing that they need to do toward the end to support their applications with the FDA. It's never just a slam dunk. They're generally a lot of work, and they're generally underappreciated how much time and money those things take.
The clinical studies at the end, what are they like?
What do you mean? Give me some more.
If you're coming from a drug space, you're used to PK, drug response. How are IVD test studies typically set up?
So you know what you're looking for, right? You're looking for specific biomarkers or morphology in tissue or different kinds of things in the blood, whatever, right? You know what you're looking for. You generally have a gold standard that you can use to be able to independently or at least orthogonally measure the same thing you're trying to measure. And then you measure it across a wide patient population that covers your indications for use and your intended use population. And you just cover all those corners, and you do it in the warm environments and cold environments. And you like pressure test all of the different environmental factors that the tests would be run in to be able to get some numbers and then demonstrate that you're able to get the same information as you would if you were doing it through the gold standard. So if you were testing something on chemiluminescence, but your system basically tests the same thing, but maybe much faster or much less expensive, or at the patient's home or whatever, you might go and sample 300 patients and have them use your thing, and then also do chemiluminescence on the same sample and make sure there's an agreement between the two. And you say, yep, we've gone through and done the work, and we've shown that our system is safe and effective, and it does what we say it's going to do. When we report out 12 units of whatever, we know that's statistically relevant and consistent across all of our systems.
Eddie, related to IVDs, what are the considerations that people we talked about clinical, but what are other considerations that are under?
The risk management side of things, those are fascinating conversations to be a part of while you're building out your risk file, your preliminary hazard analysis, thinking through URA, use-related risk analyses, thinking through your hazard matrix or your hazard analysis, and then plus the normal stuff like an FMEA, but an FMEA by itself isn't 14971 compliant. So you always got to do a bit more than that. Those conversations and the level of thinking is often underappreciated, in my opinion, when you're inexperienced in the IVD space, but you are experienced in the medical device space. Because we have to think through a bit more, we have to think a lot more through the implications of the data that we're also generating, not just would we shock someone with this thing, right? The data that generating and the data that the decisions that would be made based on that data, and the implications of all the environmental factors that are at play. A great example of that is like the level of testing that we have to do for the kind of test that would be run in a highly skilled reference lab, right? So you have a big Roche Cobas or something like that, and you're introducing a new test on that. And it's in a bigger lab. It's with really skilled technicians. They're using it, that's one thing. The next level of risk that you have to think of is say, okay, what if we were closer to the patient and we're doing it like in a POC, right? Like a proof, like
concept.
not proof of concept, sorry, point of care. So if we were doing it as close to the patient as possible, let's say in the doctor's office or maybe in the hospital, and the hospital has a smaller lab in there, but not like a big giant reference lab. Now we have to think through a bit more implications because maybe the people running the tests aren't as skilled. And then the next, you can make that even worse. And you think, well, what about home IVDs? Because there are prescription use home IVDs that you can then take home and, use it. And then there's also non-prescription home IVDs. Think of COVID tests, right? That's an in vitro diagnostic. You have to take that home, and the system that you're designing and developing has to work in the hands of a lot of different people. So now your studies get much bigger, the risks get much bigger. What's happening to this person if they get the wrong result and they actually have the thing that you're looking for, but you didn't call it correctly? Is it because they made mistakes? We have all these different use case errors that we have to think through and then we have to test against to prove that even in the corner cases of our use case population, let's say like highly skilled or highly educated versus not, and all these different corners, the system still works. We still get a good result, even in the hands of these non-skilled, non-technical folks that are like working necessarily in a hospital. It has to work for grandma, and it has to work for the high school kid down the street, and just everyone that takes the test, it has to work in. So when you think through those risks, it really helps balloon your risk management side of things, which then has an influence on your design inputs and your user needs. I think that's woefully underappreciated. It's a fun area to play in to like really think through all those implications. But once you get into those conversations and you start blowing out that side of the risk management for your IVD development, I find that's usually a lot more than people imagine. They might start with a small risk matrix and they say, yeah, we got a risk matrix. It's an Excel sheet and it's got 12 items on it. And we're like, no, you have 12,000 when we're done. We're going to think through a lot, which is a little bit different than when you have it in the hands of a skilled surgeon putting a thing inside someone, right? There's definitely risks there, but we don't need to worry about the skill level of the patient in that case.
Where can people find you, Devin?
I can be found on LinkedIn. I can be found probably easiest at Prodct, my website, which is prodct.dev. So prodct.dev is the name of the company that I help lead, and I can be easily reached through either of those two vehicles.
Awesome. Thanks for coming on again, Devin. This was awesome. We have to talk LDTs soon.